Our first speaker of the session is Chris Holmes, talking about Bayesian Boolean matrix factorization.
Thank you. It's great to be here. As everyone said so far, it's a fabulous environment. So I'm really looking forward to this week. I've really enjoyed it so far, and for the rest of the program. So what I'm going to tell today is actually a very simple story about probabilistic kind of compression, I guess, in binary matrices. So what we're trying to do is identify low-dimensional structure in very large, high-dimensional binary matrices. And when I say we, I mean, and the reason I've put this in bold, so Tamu is a PhD student at Oxford, joint with myself, and Chris Yao, who was at Oxford, is now at the University of Birmingham, and Michalis, who was a postdoc with me and worked closely with Chris, and he's now at the University of Athens. Okay, so this is very much, Tamu's work, and Tamu's currently would like to be here. He's doing an internship at Amazon Machine Learning at the moment, and he's going on to spend a year at the Turing Institute next year. Okay, so the outline of the talk. So what I'm going to give you is just the motivating example. The type of data analysis that we've been considering comes from single-cell genomics. It's quite a challenging set of data to work with. Motivated us to start thinking about Boolean matrix factorization. So you can think of this as kind of factor analysis for binary matrices. Yeah? If you know what factor analysis is, it's kind of a similar motivation. We're interested in doing it in a probabilistic setting, hence the kind of the Bayesian Boolean, if that's how we pronounce it, Boolean matrix factorization. And then, of course, the dimensionality issue, we want inference to be scalable. I'm going to present some conclusions, and really everything I talk about today is available in a paper on the archive. It's to appear in the Machine Learning Proceedings ICML later this summer. Okay. So the resolution isn't quite as good as here as it on here, but that's and essentially it doesn't matter. So this is single cell data recording gene response for a particular gene, that's a row. So for this particular gene, in about 1.3 million cells. So a lot of you would have come across talks where people talk about genomics and gene expression, and unless they've explicitly said single cell, what they actually mean is they take some tissue and you kind of fracture out the cells and you put all of the RNA and you think of it into a big bucket and you measure at the population level. But recently there's been technology has emerged which actually allows us or allows researchers to measure RNA gene expression at the single cell, at the unit of a single cell. And of course if you want to attach causal mechanisms, then we know that you have to study mechanisms at the individual level. So it's an exciting technology. It's really noisy. That's the only thing. So gene expression, for those of you thinking quite rightly, would think, well, actually that's a kind of a quantitative trait. It's continuous measurement. And the reason we're binarizing is we're just thresholding that data. It's kind of particularly noisy, so all we're going to do is we're going to transform that kind of counts or continuous data of expression transcripts into a yes-no. Is it kind of expressed, or is there evidence that it's expressed or not? So this, we have about 1.3 million cells. Those are the columns. And here we've represented 12,000 genes. Okay, so that's the data. What do we actually want to do with it? Well, we quite like to see if there's any low-dimensional structure in here. So perhaps these genes cluster onto pathways. Perhaps these cells actually as well cluster. So in cancer genomics, this has been kind of very interesting. People have realized that actually a tumor itself has quite a lot of heterogeneity. So perhaps, certain cells in a tumor have a different evolutionary path and therefore may be showing different characteristics. So that's of interest. So we want to identify the low-dimensional structure, hence the akin to factor analysis, kind of a compression. We want to quantify the uncertainty in the structure that we identify, and ultimately, we'd like to explore the correspondence, particularly in the biological data, where we actually have a way in to putative mechanisms. We would want to explore the correspondence of any underlying codes, i.e. factor load-ins, to physical mechanisms, such as etiology, disease properties. We also might like to incorporate prior knowledge. We know quite a bit about molecular biology. We might want to put that in. And we want to ensure scalable inference because the data sets are large. Okay, so those are the kind of, that's the requirements. I'm going to just talk about genomics, but just a quick flag that, you know, these binary matrices appear in other areas, topic modeling, you know, clearly collaborative filtering, where you have shoppers buy products, and we just indicate what's being bought by which individuals, and computer vision. And again, these are all in the paper if you're interested in looking. So let's actually kind of precisely define what we mean by a Boolean rather than binary matrix factorization. So we have our observed data X. This is going to be n by d, and we want to decompose it into two low-rank binary matrices. Z, which is n, so that has the same column length as X, but is L possibly, no, much less than D, and the codes U, which are D by L. And the interesting thing, or the thing that distinguishes it, is that we're going to say an observed X, which is binary, is a 1, yeah, if any of the elements, so this is a kind of an OR, if any of the elements of X, of the factors, let's call them, by the codes give a 1. Yeah? So in other words, to put it another way, the XND is only 0 if none of the factors against the load-ins are, if none of them are non-zero. Yeah, okay. So the inferred U provides a basis of L codes that compresses. You can think about it as compressing the dimensionality D of X. And the variable Z encode the presence or absence. Okay, so here's a kind of a simple example to get your head around. So here's the observed data. It's just obviously binarized digits, 0 to 9. And here's a compression, a Boolean compression. So here are the codes. So this code here just says, oh, we're going to have a code here which is a bar in the top right. This code here has 2 bars, obviously top and bottom. Yeah, and there are 6 codes and there are 9 digits. Yeah, so we've got a compression going on here. And here's the latent representation. And the black indicates it's definitely on or being used. That code is being used. The white definitely not being used, and anything in between is uncertainty. So obviously the one you want to have a look at is code number 7. So if we look at we look at the number 7, it's saying we can represent that by, well, this block here. Yep, that's present. What else is in the 7? This one here, number 3. Yep, that's in the 7. But now you can see it needs the top and the bottom. And so that's we've encoded as a 0.5 because there's going to be some error here. Because we're trying to build a 7 from this one, and then these 2 bars here. Yeah? People see that? It's kind of, all the rest are fine. You can write those as the transformation. Okay. And the Boolean part just means that we're going to represent a 2 as a kind of any of these being on, then the corresponding element in the 2 is going to be on. Okay. So that's kind of conventional Boolean factorization, matrix factorization. There's quite a bit of previous work on this. Again, I won't go into it in detail. Typically treated as a discrete basis problem with an optimization scheme to try and find those low-dimensional rank, those low-rank matrices. People have looked at using things like minimum description length to try and work out how many codes do you need to adequately represent the binary matrix. The closest work to what we present was presented last year at ICML in this paper, and they used probabilistic graphical model with a message passing algorithm to try and learn those codes. And they did a whole variety of kind of tests, and that's going to serve as a benchmark for what we're going to propose. And just to say there's some extensions, papers are present, references are present in the paper. Okay. So what's the Bayesian part? I've described the kind of logical component of Boolean matrix factorization. What's the probabilistic component? And we're just going to go for the simplest. It doesn't get any harder, by the way. This is just the simplest stochastic or probabilistic representation. So think of this as the vanilla type model, and then you can imagine how you might build upon it with hierarchies and stuff. But so now, rather than there being a deterministic x is on or not, we're going to make this just probabilistic, and we're going to make it we're going to use a logistic function. Yeah, and that logistic function just has one parameter. Yeah, it's either on with probability lambda, or it's off with corresponding minus lambda on positive lambda. So dispersion parameter lambda is positive on the positive reals, and it controls the probability of actually kind of think of it as a bit flip, yeah, in the likelihood. So here, xnd is the minimum of 1 and the inner product, the corresponding inner product of ztn and ud. Yeah, so this is the idea that you're only off if everything's off. Okay, so that is if the product related to the ndth observation in the matrix, yeah, if the corresponding factor product of that is bigger than 1, then you switch on, or the likelihood of seeing a 1 has probability alpha, which is logistic, and the alpha is greater than 1/2. And similarly, if the inner product is 0, then you are with probability 1 minus alpha, which is less than 1/2. Or to put it another way, it's bigger than 1/2 if at least one code is on. Okay. Now, clearly, you could show, simply as that lambda goes to infinity, you're going to get back to the original Boolean matrix factorization, which is all the x's, the generative model would switch all the x's on or switch them all off, depending on the corresponding factor, okay, the latent factor. Okay. We're just going to assume now that the number of codes is fixed, so we're going to fix u to some small number relative to d, the dimension of x. And we're just going to adopt Bernoulli priors on the elements of z and u. For now, independent Bernoulli priors. So this is just on the factors, think of them, and the codes, independent Bernoulli priors on them being 0 or 1. And we're going to do an iterative optimization as well for speed on the lambda. Okay. So the thing to note about this is that when we're doing the update, we can do very fast kind of Gibbs sampling or conditional updating. So just to remind ourselves, we're looking for a joint distribution. We want to define a likelihood on x, which is n by d. Z, think of those as the factors, as n by l. And u is compressing that, the d high here into l, which is low. So the full conditional for the nth element of the factor binary factor matrix has the following form. So sigma is just going to denote the logistic function, yeah, and the tilde just denotes the mapping. We're going to map it makes the notation easier. Remember I said you're either going to be logistic with plus lambda or logistic with minus lambda. So lambda is going to control the kind of the noise in the process that relates the binary inner product of z and u to the observed process. And it just, if you write it out, it just has this kind of following form. So for every element in z, you can say the full conditionals given all the other elements in z and all the elements in u and lambda has this following form. So it's a logistic with parameter lambda, this thing which maps lambda into -1, minus lambda or plus lambda, and then this sum over all the codes, yeah, the over all the all the columns yeah, or all the d elements in X. And it has this kind of little peculiar form which I will look at in the next slide. Okay, just to note that the Bernoulli prior just enters in, outside of this, and if you set, as default, if you set the Bernoulli prior to be a half, then you can just get rid of this, because this is the logit function on that, and that will just equate to 0 when you put it back through the logistic. Okay, so let's just think about sampling this element by element. Yeah, we're just going to do element by element through Z and through U. Okay, so we're looking to sample this Z, and the thing to note is that it allows fast updating, as under certain conditions the argument is known without considering all of the remainder of the Markov blanket. So you just need some checkpointing through the algorithm just to ask two conditions. Well, first of all, there's one condition. So if you look at this sum in here, this says if I go to update the nth element of Z and how that equates to the data, it equates to the nth row of X. Yeah? So we're going to have to sum over the observations across the D, the D elements of X on the nth sample and their corresponding codes. Yeah? So the first thing to note, if LD is 0, there's nothing more to be done. The other kind of interesting thing is this product, is over all of the other, over the L factors. Yeah, and because of the kind of Boolean nature of the model, is that if at least one of them is 1 on some of the other codes, then you're done. Yeah, because the reason for that being is that you break the dependence between the particular ZL and that X. Okay, so because of the nature of the Boolean logic, if one of those codes is already kind of on, then flipping the Z between a 0 and 1 will have no effect. Doesn't change the probability, the likelihood of observing that particular X. So you get fast checkpointing essentially inside. So how can we do updates? Well, the updates can be just made using a metropolised Gibbs sampler, which just means we're going to start the sampler off from a particular binary realization of Z and binary realization of U, and we're just going to flip the bits. Yeah, so you're going to pick one out. Well, we're going to sweep through in a random order, choose a bit, propose to flip it, accept or reject with based on that particular probability, and similarly for U. Okay. It's quick because the checkpointing, and this is just a standard like logistic. Yeah, so it's a logistic, and it only takes 2 parameters, plus lambda or minus lambda. So for the single cell data that I showed earlier, we have around 10 to the 10 observations. Okay, so we're trying to do this matrix compression on 10 to the 10 observations, and if we compress that down into, say, 2 latent factors, that takes around about 5 minutes to do a full sweep through the Gibbs sampler, and that's on a 24 computing core. Okay. The dispersion parameter, for now, we're just going to maximize it. So obviously, you could think, well, we could put a prior distribution on it, and you could also update it as part of the process. We're just going to, as I said, for now, we're just going to try and kind of hill climb on it. And the way we do that is just given the current values of the codes U and the latent Z, we're going to look at the error rate. Yeah? So we just look at the reconstruction error rate. So all this says is, let's look over all elements for XND, and let's just count whether the map prediction, whether you were above a half if the X was a 1, or whether you were predicting it as less than a half if the element was a 0. So this is just going to count the error rate, and that allows us to then write the likelihood function yeah, as a function of lambda. And then obviously trivial maximization of that says, you know, this is why we can think of lambda as the overall kind of error rate. It says, well, how well are you doing when you compress down to L codes? Yeah, how well do you do in reconstructing X? And that's just the kind of the overall error rate. And hence we're going to alternate between sampling full sweeps through U and Z, and then maximizing lambda until convergence. Yeah? So by convergence, I mean it takes 5 minutes to do one sweep. So essentially, we would typically do, like, say, 100 sweeps through. Although on the experiments we've looked at, the results are pretty stable under that. Okay. There's another thing that you can play with, which is a multi-layer construction. Yeah. So as a kind of an obvious generalization is now to kind of stack up these latent representations. So here is just one example where we've taken 50 digits and we've added noise. So we've randomly flipped bits to make those images noisy, and then we've written this as a primary layer which is reconstructed from factors or codes at a lower level which are reconstructed from factors at a lower level. Yeah, so we go from 2 codes to 4 to however many they are there. And what we've seen when you do that, again, it's very fast and it's probabilistic, is that on this particular, it's only just one kind of example, but you improve the reconstruction error, or rather for the same number of total codes, you get better compression. You seem to get better compression. So the reconstruction error on these kind of 50 digits was went from 1.4% when we were doing a single layer Boolean matrix factorization down to 0.4. We'll see in the single cell example when we come back to that towards the end of the talk that actually independently running these under hierarchies you get quite a lot of correspondence between the codes at different levels. Okay, so here's some experiments. So we generated a square matrix n by n of rank L by taking Boolean product of 2 lower dimensional Boolean matrices, n by L. Okay, so this is square. So both Z and U are going to be n by L. Okay, and if we take just bit flips within the codes and say the factors, then we know the expected value of X. It has this formula here, and we're gonna do that such that the expected value of X is 0.5. Yeah, just going to do that. So the overall amount of zeros to ones in the data, just for this simulated example, we're gonna specify that, and then we compared our approach with the kind of message passing approach of Ravenbakers, who in ICML on the previous year, last year. Okay, and this is kind of typically what we see. It's kind of an interesting. So this is the reconstruction error from simulated data on an n by n, so on a 1,000 by 1,000 matrix. Here's the average reconstruction error by the amount of bit flipping we do. Yeah, so we generate data, and then we flip bits at a 5% automatic flip up to a 45% of the data flips. And we took the rank as 5. So the intrinsic dimensionality of the data is 5. And I guess we repeat this 10 times and look at the overall error. And so what we can see is that this kind of OrMachine, or this Bayesian Boolean matrix factorization gets very good reconstruction. I mean, it's a kind of a nice data set for it to look at. Yeah, it's very low dimensional, highly structured, but we're getting exceptionally good reconstruction up to about the level of 40, and then something happens and it just off it goes, yeah? So then this big jump. And just to say, those error bars are based on 10 repeats of the data. Okay. So let's lower it down. So we now did a smaller matrix. So now this is 100 by 100, and the rank's gone up to 7. Yeah, and again we see similar kind of performance. So there's a boost in performance from using the kind of, in some sense, the fully or more probabilistic. Yeah, is that such a word? Using a probabilistic version with a Gibbs sampler rather than a message passing version. And again, we can see that on this example, we're outperforming the message passing. And it's kind of we looked at it. So here, not only are we showing reconstruction to the truth, we're showing reconstruction to the noisy observation. Okay, and so the Bayes error is the line y equals x. So if I take this low dimensional, if the truth is a low rank of rank 7, and now I randomly flip bits at a rate of 15%, you can't do better than getting a 15% reconstruction error. Well, that's the Bayes risk, associated with that reconstruction classification. And so what you can see is that both message passing and our approach is kind of tracking the Bayes risk at about 20% reconstruction error. That gives us at around 20% flipping. But then as you go forward, it looks like, we haven't really done the diagnosis, but this is consistent with the message passing doing a little bit of overfitting in this portion here. Because we know the Bayes risk is 50%. And if you just looked at the error on the kind of training data, it's below 40%. Yeah. So I think that the message passing is probably overfitting a little bit in these kind of noisy areas. And I guess maybe one reason for that is, of course, the message passing is taking a single mode. Yeah. So it's passing to a kind of a single model, whereas we're doing some averaging over Bayesian model averaging. Okay. We looked at various things, so then we did kind of model misspecification. If we set the priors and the parameters that say we expect 50% zeros to 50% ones, but then we generate data with a higher expected value, what happens? And it looks like that there's some more flexibility in the fully probable, as you'd expect in the fully probabilistic approach, to be able to accommodate a mismatch between the prior and, say, and the data generating process. And we also looked at this issue, this kind of interesting problem about matrix completion, which is if we take X and we remove certain elements. So now you've got unobserved values, and what you want to do is you want to fill the matrix in. Okay, so X binary matrix, you've got missing values in there, you want to predict or you want to complete the matrix. And so again, you could use these methods there. When we look at, this is now the correct number of elements explained, we see that we seem to do better. When actually we partition the predictive distributions for things we get correct and things that we get incorrect, it has these two forms. So when in a sense, when we're correct, we kind of know, or we suspect we're correct. So here's the predictive distribution, the blue line, for the things we get right. Yeah, and of course, because there's a lot of certainty here. You're making certain predictions. The things that we get wrong is the green, and so there you can see that actually the model is, if we're going to anthropomorphize it, the model is much more uncertain in those predictions that it gets wrong. So there's a kind of interesting look at the calibration issue there. So what do we do how do we do on single cell analysis? So this is going back now to the mouse data that we have. So we applied this to the binarized gene expression data of about 1.3 million cells. There were about 28,000 genes, say, per cell. We actually kind of reduced that down to about 11,000 or so. So for genes that were showing reasonable levels of expression. So the cell specimens are obtained from the heads of mice, of embryonic mice. So this is cortex hippocampus of embryonic mice at day 18. Okay, and just to note, this kind of data is fairly sparse, is that 7% of the data, there's only 7% kind of non-zeros here, which means that for any cell, on average, 7% of the genes are kind of expressed at this when we come to threshold it. Okay, so I said we remove genes expressed in less 1% of cells. That leaves around 10 to the 10 data points in the matrix. And what we did is we applied, we took the number of codes and we increased it sequentially from the results I'll show are from 2 and 3 and 4. And the thing to note is we did that independently. So there's no kind of, for this example, we didn't do that in a hierarchical setting. We just ran the method by saying, right, you've got 2 codes, compress it down into 2 codes, compress it down into 3 codes. We ran about 100 samples, or the results here are based on about 100 posterior samples. Takes around an hour, about 30 minutes on a cluster with 24 cores. And then what we then did is we did some analysis on the codes that were identified. And so, what we're interested in saying is, the task is that if we're going to compress this high-dimensional data down into low-dimensional codes, do they represent anything of interest? And to do that, we use enrichment, standard kind of enrichment tools in bioinformatics. So essentially, it just says, are these particular compressions enriched for certain genes in certain pathways? Okay. So here's a kind of result. It's a bit busy plot. So if I go through, if you go through kind of row by row, so at the top, we're showing when we run it with 2 codes. So we're going to compress that down, that U matrix now. We're going to compress down in just the kind of 2 codes. And each bar here, all it does, this is not kind of related to the elements, it just says whether a particular gene, so there's a gene per bar, whether it had a code that was on or not, in this particular factor, for this code, whether it was switched on or not. And so, and we've kind of ordered the genes in a particular way just to highlight how many genes are kind of present or covered by particular factors. I don't know if you've got the resolution to see. We can also look at enrichment for particular pathways as the codes. As you add more and more, you do less and less compression. And so kind of unsurprisingly, we've got things like olfactory bulb and neurological signals or expression pathways that are represented in this particular data. There's another thing that we tend to see is that you tend to see stability between the factors across as we add more, and that's unusual for those of us that have worked on factor analysis. As you add more factors, you can often see quite different things emerge. I think here, because of this kind of the Boolean nature or the OR nature, means that you tend to get this kind of strong correspondence between the codes run at different levels. Okay. So here we can look at the kind of column enrichment that we've ordered by display, and we can look at how the kind of genes are divided up or represented by particular pathways or particular codes as we add more and more codes into the model. The other thing we can do is we can do the same for the individual cells. Okay, and this would be interesting in particular if there's heterogeneity in a tissue. So as I said in in tumours, if you take if you do a micro dissection of a tumour, you could often expect to have very different mechanisms present in there, because we know that tumours are quite heterogeneous in terms of their kind of evolutionary background, local evolutionary background. So here we kind of again play the same game where we can now, the columns here represent each one of the 1.3 million cells, and we can look at their presence, how you allocate cells to particular codes for evidence that there's clustering in that sense. And again, we can see that there seems to be strong clustering against the things of interest. Okay. Conclusions. So just one slide of discussion points. We've developed a kind of very vanilla version of Bayesian. It's a probabilistic Boolean matrix factorization. Said the paper's there. We searched around. We think it's about, couldn't really find much other stuff out there on probabilistic, or fully probabilistic in a sense, or more probabilistic, if that's the word, to perform posterior inference on Boolean matrix factorization. This checkpointing allows for very efficient sampling using Metropolised Gibbs and just flip in bits in the Z and in the codes U. What we've seen is that multi-layer networks improve compression. So for the same number of codes, you can do greater compression. There's obvious kind of extensions to take this to. At the moment, we just have one single parameter, lambda. So you can imagine that we optimize. So you can imagine putting a prior onto that. You can imagine trying to do some hierarchical things. Perhaps some non-parametric priors, and an underlying kind of restaurant process that would generate this. And in a sense, that would accommodate the uncertainty in L. Yeah, so at the moment, we're kind of deterministically running through L as sweeping through L is 2, then rerunning for L equals 3. You can imagine it could be interesting to say, well, what's the evidence in the data for a certain level of underlying compression. Just to acknowledge EPSRC funding through ILIKE, Medical Research Council grant, Program Leaders grant for me. Thank you. And that's a little logo that Tamu did. Thanks.
We have lots of time for questions.
So the computing was done on a cluster of 24 cores, and it looks like each machine was heavily used. I mean, it takes 30 minutes or so to do the analysis. Any reason you chose a cluster instead of a GPU? Because it seems like 1,000 cores would do better on something like this.
Yeah, it was just, I think it was local hardware that was available. Yeah, that was the only reason. Yeah, for that. Hey.
What if you, like at the moment you've just got one, like it's only one, if one is on then everything's on, and if, so, I mean I understand the logic in some problems, but how does that generalize? Like what if it's more than that, or do you need a threshold? I mean then it becomes something like,
Yeah, I mean the thing is to in some sense respecting the Boolean logic nature of the original kind of the matrix factorizations, which just has this OR. Yeah, if any of the codes are on, then you're represented as a 1, and you're represented as a 0 if none of the codes are on. Yeah. So yeah, I mean there are probably well, I imagine there's other things. Mappings that you could do, non-Boolean mappings, but that was the way that we were trying to respect that.
You have time for more questions?
Is it a bit like a stochastic EM, more or less, what you've done? it's reminiscent. I mean,
Yeah.
it's back, but it is reminiscent. It looks
Yes, this idea as well of like optimizing. I mean, the things, you know, that are some, you know, computational hacks in there, like this optimization of the lambda. We played around a bit, or Tamu rather, kind of played around a bit. I mean, the interpretation of that lambda is exactly the kind of the noise level. It kind of represents the intrinsic noise of the process. And so we again, as I said, we just optimized it, but it's clearly not that, what we've done is not kind of strongly motivated in a fully Bayesian approach. But yes, it's a bit like an EM. It would be an EM, wouldn't it, if we could get the marginal likelihood. It would be type 2 Bayes if we could get the log marginal likelihood and then maximize the lambda at each step. And we're not doing that. We're just maximizing the lambda given the realization of the U's and the Z's.
More questions?
Thank you.
Our speaker again.